Now, DNA replication takes place in all living cells. All organisms, whether they belong to the bacteria or the archaea or the eukarya domain, they have DNA as their genetic material. Remember, that's part of our definition of a living cell. So they're all performing DNA replication, and they're all doing it in basically the same way. There are some key differences, though, and we should be aware of those. We're going to use the prokaryotic process of DNA replication as our model as we talk through the step-by-step process. There are differences, again, between the way prokaryotes do it, bacteria and archaea, and eukaryotes do it. I'm going to point out to you where those key differences are. Now, there's something else to mention at this point, and that's this. DNA polymerase enzymes are numbered. So when you read about prokaryotic and eukaryotic DNA polymerase enzymes, you'll see that they're all given numbers. DNA polymerase 3 or DNA polymerase 1. DNA polymerase enzymes perform different jobs in the cell. Some of them perform more than one job. And if you recall, when we talked about enzymes, that's unusual. Normally, we expect an enzyme to do one job, catalyze one reaction, and do it very well. DNA polymerase is a little bit different. Depending on which form of the enzyme you're talking about and in which kind of cell, prokaryotic or eukaryotic, it can be performing different jobs. For example, the DNA polymerase 3 enzyme performs DNA replication, but it can also perform proofreading. In other words, after the new DNA is synthesized, that same enzyme can do the job of proofreading the new strand, which means going across the new strand and making sure that all of the base pairings are correct. And that enzyme, if it finds a mistake in the base pairing between the new and the old strand, it can repair that mistake. So this is a pretty amazing enzyme in terms of its abilities. So we're going to talk about this process of DNA replication step by step, and we're going to talk about it the way it happens in a prokaryotic cell. DNA replication proceeds in two directions from what we refer to as a point of origin. Now, in some reference materials, you'll hear them refer to it as the origin of replication. The idea is the same. There's going to be some point, some location in the double-stranded DNA molecule that will be the place where DNA replication begins. And the very first thing that needs to happen is the double helix needs to be pulled apart. It needs to have the two strands separated from each other. This is going to create what we call a replication bubble in the chromosome. It's just an opening between the two strands. In prokaryotic cells, there's going to be one bubble because remember, prokaryotic cells have a circular chromosome. In eukaryotic cells, it's different. In eukaryotic cells, because there are multiple linear chromosomes, you're going to find multiple replication bubbles. Not just one on each linear chromosome, but several on each linear chromosome. So that's one of these key differences between prokaryotic DNA replication and eukaryotic DNA replication. Now, this replication bubble, when it first forms, is quite small, but as DNA replication proceeds, that bubble gets bigger and bigger and bigger. It opens up in both directions. DNA synthesis, remember, can only be done in the 5' to 3' direction. That means that the DNA polymerase enzyme can only build DNA in that direction, 5' to 3'. It can only add new nucleotides to a growing strand, a new strand of DNA at the 3' end of the molecule. What you're looking at the bottom of this slide is an image taken with an electron microscope. You're looking at a portion of the circular chromosome in a prokaryotic cell. So this is double-stranded DNA over here. This is double-stranded DNA over here. And this is a replication bubble. So the point of origin would have been somewhere right around here. These are two individual strands of DNA, two old strands of DNA. They've just been separated from each other. They're connected over here still. They're connected over here still. But we have opened up this bubble right here. This bubble is going to get bigger and bigger and bigger. It's going to open up in this direction as we separate these strands more. It's going to open up in this direction as we separate these strands more. Bidirectional DNA replication. And here's a drawing. You can see this is a chromosome from a prokaryotic cell. It happens to represent a bacterial chromosome. It's circular. And right here, a replication bubble has been opened. So right in the middle of this, there was a point of origin, or again, what we sometimes call an origin of replication. The two strands were separated at that point of origin. Now, what they've drawn in red is new DNA being made. The DNA polymerase enzyme is coming in and it's making some new DNA. New DNA is made on each of the old DNA strands. Now, as the new DNA gets made, we have to provide it with more templates. So we have to keep opening up the bubble. And that's what they're showing us here. The bubble has gotten a lot bigger over here. You can see new DNA being made and new DNA being made. Notice that they're showing us that the arrow on the new strand, the red strand, is pointed this way. And the arrow is pointed this way. Two different directions. And that's because the old strands of DNA are anti-parallel. So because we can only build DNA 5' to 3', you're going to see it occurring in different directions on the two old strands. Now this is a drawing that represents a eukaryotic linear chromosome. And you can see there's not one replication bubble here, there are 3 drawn here. And that's what we see in eukaryotic cells, multiple replication bubbles on these linear chromosomes. Now these look pretty much identical to the one you saw for the prokaryotic cell. But there is a new term introduced on this slide, and that's the replication fork. This is a fork right here, and this is a fork right here. So a replication fork is the junction between where the DNA is still double-stranded and where we've separated into 2 individual strands. These are called the replication forks of the replication bubble. So the action, if you will, of replication happens at the corners of the bubble, at these forks. Okay? Those forks are moving. That bubble is continually getting bigger and bigger and bigger because we need to provide the enzyme with more template to build new DNA on. So the bubble gets bigger and bigger because the forks open. Now, there are very particular proteins in the cell that are responsible for making all of this happen. And that's probably no surprise to you at this point. These proteins have to be able to recognize these particular sites and assist in this process of replication. Obviously, a lot of these are enzymes, but there are also other proteins that are not enzymatic, not catalytic, that are going to help do this work. So first things first, we have to locate the point of origin, and we have to separate the two strands of DNA. And not only does that have to happen, but we have to stabilize this. We have to keep the two strands separated. Remember, they're attracted to each other. Those two strands of DNA want to hydrogen bond with each other. So we have to stabilize them separated. So an enzyme called helicase comes in, and it locates the point of origin, and it breaks the bonds between the two strands of DNA. In other words, it is the first enzyme to come in and unwind or separate that double helix. Once the two strands are separated, even a little bit, some of these accessory proteins have to come in and help. There are what we call single-strand DNA binding proteins. That's a name that makes a lot of sense, right? Single strand, they're going to come in and they're going to bind to the separated strands of DNA, and they're going to sit there, these proteins. And the reason they do this is because they will be a physical block to those two strands coming back together and reforming a double helix. Now, there's a little bit of a problem when you open up a double helix of DNA, particularly a circular double helix of DNA. And you have to use sort of your imagination to visualize this. When you pull apart those two strands, what you're going to do is cause the double-stranded areas that are outside of the replication forks to wind around each other even tighter. What this is going to produce eventually is an excessive amount of coiling and then kinking in the double-stranded portions of the chromosome outside of the replication bubble. And I'll show you a drawing of this in a minute that might make it easier to visualize. If a cell opened up a replication bubble and then kept opening it up, what would happen is the DNA that was still double-stranded outside of the replication bubble would become so coiled and so kinked that it would get damaged. So a second enzyme has to come into the picture here. And this is actually a family of enzymes called topoisomerases. Topoisomerases are enzymes that can cut the DNA helix, double helix, and then rejoin the broken pieces. And they act outside of the replication bubble. So imagine if you had two pieces of rope that were wrapped around each other, and you twisted them and twisted them and twisted them until they started kinking up. If you cut one of those pieces or both of those pieces of rope, all of that built up tension would be relieved. All of that twisting and kinking would go away. That's what a topoisomerase enzyme does. So this drawing is trying to show us that visually. Here is a double helix of DNA in a bacterial cell, a circular chromosome. And you can just see that one of the strands is red and one of the strands is blue. Now, when we begin DNA replication, we're going to separate those two strands at the point of origin and create a replication bubble. And then that bubble is going to open up here and here. It's going to get wider and wider and wider. As it does that, it causes this. As we unwind one part of the circle, we wind the rest of the circle even more tightly. You start to see not only overwinding, but actual kinking of the double helix, this is something you would not want to allow as a living cell, because this could damage this very precious DNA molecule. So in come the topoisomerase enzymes, and that's what we're looking at on this slide. They're showing us a piece of double-stranded DNA up at the top of the slide, and then they're showing us that same strand as we try to open it up. So this isn't a drawing of a complete replication bubble. If it were a complete bubble, it would be open here, and then it would come down here like this, and there would be more double-stranded DNA over in this direction. So we're looking at sort of half of the replication bubble here. As the bubble separates the strands, as it takes away the coiling here, it adds more coiling over here. This is referred to as supercoiling. And if we allow supercoiling to get too extreme in a cell, it will kink and damage this DNA. So in comes the topoisomerase enzyme, and all it has to do is make a little nick in this supercoiled DNA, and it will sort of spin out all that excessive tension. Then it just seals it back up. And as the bubble gets wider and wider and wider, it does it again. It nicks the DNA, lets all that tension out, and then it seals it back up. Topoisomerase enzymes. Now, one of the most confusing things about DNA replication is what I'm about to talk about next, and that's this concept of leading strand and lagging strand synthesis. There are actually two functional regions inside a replication bubble. There's what we refer to as the leading strand regions and the lagging strand regions. And that's because the cell is going to make new DNA a little bit different in these two areas. So each of the new strands of DNA that are being made, remember there are two old strands of DNA, and each one is being used as a template. So there are two new strands of DNA being made. Within each strand of new DNA, there's going to be a region where the DNA is made using this leading strand process. And then there's another region that's going to be made using what we call lagging strand process. Okay, makes sense? All right, let's take a look. This is a drawing showing us these regions in a replication bubble. Here's the double-stranded DNA over here. Here's replication bubble. Here's double strand over here. There's a replication fork right here. There's a replication fork right here. Point of origin, right here. We pulled the two strands apart here with the helicase enzyme. Single-stranded binding proteins came in and helped keep it open. Topoisomerase enzymes are ready to relieve pressure out here, outside the replication bubble. DNA polymerase is going to use the old strands to make new strands. We're going to make a new strand here, and we're going to make a new strand here. And because the two old strands are anti-parallel to each other, one of the new strands will be made in one direction, and the other new strand will be made in the other direction. Now look at each of the new strands. A portion of each new strand will be made using what we call leading strand synthesis, and a portion of the new strand will be made using lagging strand synthesis. And right away you can see that the difference visually is that the leading strand areas are continuous. They are one long piece of new DNA, and the lagging strand areas are fragments of new DNA. Those fragments get a name. We call them Okazaki fragments, named after the person who first described them. Leading strand replication is a continuous form of replication. The enzyme is just adding new nucleotides, one at a time, one after another, after another, after another. The way that the leading strand area is oriented is it has its 5' end at the point of origin. It has its 3' end at the replication fork. So that means it's adding new nucleotides at the replication fork. As each new dNTP comes in, it will be added at the 3' hydroxy end, like all DNA is made. And the 3' hydroxy end just happens to be located at the fork. Let's take a look. Here's the leading strand, or what's being made in leading strand synthesis. So the enzyme comes in, it adds one base here, one nucleotide here, one nucleotide here, then another, then another, then another, then another, and then as the bubble gets wider and wider and wider, it just keeps adding new nucleotides right here, right where the fork opens up. It's doing the same thing on this strand. It's adding new bases, new nucleotides at the fork. As this gets opened up more and as new template is exposed, the enzyme just adds the complementary base to each exposed old base, and it builds new DNA at the fork. Now look at the leading strand, the lagging strand areas. These are oriented differently. They have to be oriented differently, right? This strand, this new strand of DNA, all has to be oriented in the same direction, 5' to 3'. It's just that if you go forward from the origin, that's leading strand. If you go sort of backwards from the origin, that has to be lagging strand, because the enzyme is adding new nucleotides to this end. So the bubble opens up a little bit, the enzyme starts here and builds new DNA. The bubble opens up a little bit more. Remember, it's opening up over here. What does the enzyme do? It has to literally jump back and build DNA this way. And then it has to jump back and build DNA this way. And as that bubble opens up more, it will jump back again and build DNA this way. And then it will jump back again and build DNA this way. So because DNA can only be made in one direction, there will be a portion of each new strand of DNA that's made continuously. That's what we call leading strand synthesis. And there will be a portion that is made discontinuously, or in fragments. That's what we call lagging strand synthesis. The lagging strand area is a discontinuous form of replication, because now the new strand has its 3' end at the origin of replication and its 5' end at the fork. Exactly the opposite of the leading strand areas. So new dNTPs are added not at the fork, but away from the fork, toward the origin of replication. The enzyme has to jump back and build forward.